Apparently, I'm supposed to self-introduce myself. I'm George Bush. Now, I understand I don't want to really offend any Republicans in the audience. This is meant to be an evolutionary biology slide. And as Kimmin pointed out to me, I'm not going to tell you which one I think is the missing link in this slide. So if you looked at the title of my talk, it's the axis of evol, which I really feel like talking about all the time, because currently the use of evolution and even the belief in evolution is unfortunately a bit of a controversy. And what I'm going to talk about today is basically applied uses of evolution in metagenomic analysis. And the basis for this is basically a quote by Dobzhansky, a famous population geneticist of the 20th century. And he wrote this quote actually in an article on science teaching. And his point of this was that you can't understand, in particular, things about comparative biology without understanding evolution. And he wanted to distinguish comparative approaches, which people identified similarities and differences among organisms or among genes or within populations, from trying to figure out how and even why those similarities and differences came into being. So it's a fundamental different perspective, comparative, determining the differences and similarities, to trying to figure out how and why those similarities and differences came into being. And there's a plethora of examples from molecular biology where evolutionary approaches have been fundamental in interpreting and understanding certain molecular phenomena. And I'm not going to go through all of these. One of my favorites, which is of relevance to the crowd tonight, is that Robin Gattel and colleagues were able to basically solve the structure of the ribosomal RNA molecules from looking at correlated evolutionary substitutions along evolutionary trees that let them figure out what the secondary structure was of the molecules. And that was not possible to do that from de novo predictions of structure using folding algorithms or other methods. It only came from an evolutionary approach. And there's a plethora of these examples. And for better or worse, many years ago, I invented my own omics word. I apologize for that. And it's phylogenomics. And what I meant by this was basically that there are many ways that evolutionary approaches can benefit genome reconstructions, and there are clearly many ways that genome analysis can benefit an understanding of evolution. And really, this should be an integrated approach where you combine the two things into one analysis. And what I want to talk about today is combining metagenomics and evolution. And I tried to come up with a word. I'm sorry, I wanted to invent another word. Joe Handelsman came up with this term metagenomics, and she sent me her paper, and I looked through it. I don't think she really described the whole root of the word metagenomics. She said it was okay to make fun of her when she was gone. So, and I still don't understand exactly what metagenomics means, so I figured maybe meta-evolution. And so I Googled, of course, meta-evolution, and I came up with this as the top definition. Try and read this. I have absolutely no idea what. In meta-evolution, the vertical transition to an adjacent level may be understood as symmetry break, which implies an increase in complexity. I won't even go on with it. I mean, it's just completely baffling. So of course, I went to register.com and tried to register various domain names, and this is what it suggested as alternatives to metagenomics. Some of these are great. My Metagenomics I really like. Metagenomics Live. But of course, Euro-metagenomics, that's for the Europeans in the crowd. We're not neglecting them here. And of course, hot metagenomics. But none of these really applied. So unfortunately, I came up with Phylometagenomics. It's not as catchy. But basically, what I'm going to argue is, just like with phylogenomics, there are many ways where evolutionary approaches can be combined with metagenomic analysis, just like with genomic analysis, into an integrated field. And since I added this slide in the discussion where we're supposed to talk about our question, I'm going to give you some examples of this. But what I'm personally interested in scientifically is the origin of novelty, where new processes and functions, how they come into being, the mechanisms by which they come into being. And in particular, recently I've been interested in a very simple way that organisms can acquire new functions, which is they can acquire them from other organisms, either from other relatives within their group by recombination, or by lateral gene transfer. We've heard examples of that here, or through symbiosis. So you can just work with another partner and take the functions from that partner. And so what I'm going to do is take you through 3 examples different areas where evolutionary approaches can benefit metagenomics. Most of these you've already heard about at this meeting. I'm going to sort of reiterate them with some of my own examples and just try and bring the point that evolutionary approaches are very beneficial in metagenomic studies. So I love this from the Weekly World News, which if you want to get good slides for evolution talks, just go by the Weekly World News any week. And they have amazing stuff in there. This is to remind me that one of the tasks in metagenomic analysis is trying to figure out who's out there. And of course, they figured out that this is the flu bug here. I think they may have made a mistake in this. But of course, we know that people haven't talked about it here explicitly as an evolutionary approach. But phylotyping, the thing started by Norm Pace with characterizing uncultured organisms using the ribosomal RNA tree, is explicitly a phylogenetic approach. And most of the people practicing phylotyping still use phylogenetic trees to take ribosomal RNAs from uncultured organisms and assign them to a position in the tree, and thereby get an idea of what type of organism that is. Now, there are people who don't use phylogenetic approaches to do this phylotyping, blast searches, et cetera. It's better to do a phylogenetic approach, but actually most people in this crowd use ARB or their own system to do a phylogenetic typing of the ribosomal RNAs. And one of the reasons this is better than, for example, a blast search with your ribosomal RNA is that phylogenetic trees group the genes by their relatedness. Blast searches group things by their level of similarity. And the level of similarity is not always a perfect replicate of your relatives in an evolutionary analysis. The best example of this is convergent evolution, which makes similarity be misleading. So for metagenomic analysis, of course, you can do the same type of thing that Norm and colleagues developed for PCR. You can take the ribosomal RNAs from environmental samples and do phylotyping with them. So this is from the Venter Sargasso C paper, where we took all of the reasonably sized ribosomal RNA sequences from the Sargasso metagenomic data. So now we have a non-PCR-based approach to get ribosomal RNAs. Now, you might call this unbiased. It's got different biases than the PCR approach. And we did the same type of thing. We classified each of these ribosomal RNAs by building trees of all of them. And we can take that information and get a picture of the phylotype diversity from the sample. You've seen examples of this from other talks here. This is basically the same idea, something we did for the Sargasso C paper. And we've assigned them, for example, to major phyla or taxonomic groups based on their position in the ribosomal RNA tree. And this gives you a picture of the phylogenetic types without the biases of PCR from an environmental sample. Something that actually hasn't been talked about explicitly at this meeting, although Ed used this figure in his talk, is that the studies of diversity using ribosomal RNA, like Chow diversity indices and any measure where you're looking at operational taxonomic units or OTUs or something like that, most people do that with a similarity-based approach. So just set a cutoff threshold of 97% identity, and that defines your operational taxonomic unit. A variety of people, including Andrew Martin in a really nice paper, have pointed out that it's probably better to do a phylogenetic approach to defining operational taxonomic units, where you set various cutoffs in your tree rather than as percent similarity, and then use those levels in the tree to define operational taxonomic units. And you do get somewhat different pictures of the diversity of a sample than by setting just percent similarity cutoffs by doing this. It turns out you can actually do all sorts of interesting ecological and evolutionary analyses where you take this phylogenetic approach rather than a similarity-based approach. So also in this paper by Martin, talks about how you can use a measure that's called FST. It's sort of a population genetics measure of differences within and between groups of individuals. So for example, if you had a tree of all the ribosomal RNAs from a particular study, and you looked at the diversity within this, if the white genes here were from one sample, if you pull those out of the tree of everything, the tree would look like this. If the black ones, you pull them out of everything, the trees are identical, actually, basically, in terms of their topology, even though the ribotypes are not identical. This is very similar to what Jeff Gordon was talking about with the human gut studies, where you get somewhat different subtypes within some individuals, but the overall picture of diversity is similar. This would be, rather than cataloging the phylogenetic types, this would actually be taking a truly phylogenetic approach to quantifying and measuring the diversity within the samples. Of course, as we heard in our discussion earlier, we're overwhelmed in some ways with metagenomic data, so we have to figure out ways to do these analyses as much as possible in a high throughput manner. And for the people in my group, Danying Wu in particular, developed a pipeline that we used for the Sargasso study a little bit, and more so for the new Global Ocean Survey papers and some other work we've done. That's an automated phylogenetic tree building algorithm for ribosomal RNAs. There are other methods that people have described out there that are similar to this. This is our own. Basically, we do an iterative search. We take a ribosomal RNA, we build a tree of it to figure out whether or not it's a bacteria, archaea, or eukaryote. Then we zoom in on that group and we assign it to a phyla, and then we zoom in on that phyla and assign it to a particular sub-taxonomic group. But it's taking not a blast-based approach, but a phylogenetic approach to classifying ribosomal RNAs from environmental samples. The great thing about this, even though it's got its imperfections, it doesn't always do everything correctly probably, and if you curated the analyses, you might get a little more precision. But we don't have time to do curation in all of these studies. So with 36 or 35 of the GOS samples, we took all of the ribosomal RNAs in those and ran this algorithm on them. And the bars correspond to different phyla in the samples. And now we can get a picture of the changes in the relative frequency of ribosomal RNA sequences from environmental samples across these different types using this phylogenetic approach. And it's still much more computationally costly than doing a blast-based classification of sequences, but I think it's sufficiently more accurate in terms of the taxonomy that it's worth trying to do. To me, the greatest thing about metagenomic surveys is that we can go beyond ribosomal RNA to looking at other phylogenetic markers in environmental samples. And there have been, ribosomal RNA has single-handedly, helped revolutionize environmental microbiology. But there are a lot of, subtle issues with relying solely on ribosomal RNA sequences to assign information about the taxonomic diversity in a sample. We can talk about those afterwards if you want. But there are a lot of reasons why it would be worth looking at other genes. And so in the Sargasso Sea study, we took my favorite phylogenetic marker, RecA, and did the same thing that you would do with ribosomal RNA now with RecA. We'd never been able to do this before because degenerate PCR for protein coding genes simply doesn't really work. And you don't get anything near as universal as PCR for ribosomal RNA. So you can't clone the RecAs from across any large taxonomic group from an environmental sample, despite that RecA actually has some really nice sites for PCR primers. It has some amino acids that are nearly 100% conserved that are only one, twofold or not even degenerate amino acids. So, M, K, Y, K, N. You'd think they'd be great PCR primers, and we've tried it in environmental samples, and it's just not robust like ribosomal RNA. But now we have this random shotgun data, and we can go through it and find all the RecAs and use them to do taxonomic classification just like with ribosomal RNA. We did this with 5 protein coding genes for the Sargasso Sea paper and compared them in what we think is a relatively fair comparison with ribosomal RNA. The reason I say fair comparison is the ribosomal RNA database is, of course, gargantuan, and the RecA database and other protein sequence databases are not. So we actually took the sequences of these genes only from completed genomes and did the phylogenetic analysis just with that set so that we could compare RecA to ribosomal RNA to these other genes and see what answers they gave. And so, for example, all of the genes said that the alpha proteobacteria were the most abundant phylotype from those that in the Sargasso Sea data set. All of them said that the second most abundant phylotype were the gamma proteobacteria, consistent with ribosomal RNA PCR studies. What I think is the most interesting thing that came out of this, and we are following this up with some other simulations and some other genome analyses, but you can actually see it even in this, the first analysis that we did, which was in the Sargasso Sea paper, is that the relative proportion, so the quantitative picture is slightly different among some of these genes, although the qualitative picture is similar. So if you look at just the gamma proteobacteria, for example, all of the protein coding genes say the gamma proteobacteria corresponds to about 20% of the sequences that we found, whereas the ribosomal RNA says it corresponds to about 30%. And there are other differences among the markers. Frequently, especially in the new analyses we've done, the protein coding genes tend to agree on their quantitative estimates more so than they agree with the ribosomal RNA. I think this is due to different copy numbers of the ribosomal RNAs within genomes. So in prokaryotes, you can have up to 15 copies of the ribosomal RNA in a genome. If you sample another ribosomal RNA from that one cell, you might count it as a second sample, even though it's a single cell. Whereas most of these protein coding genes are present in 1 or 2 copies. So the variance in copy number is much lower, and therefore they are better markers for looking at relative abundance of organisms from metagenomic data than ribosomal RNA is. They're just, in principle, they are better. And they're useful phylogenetic markers, so you can do the same types of analyses as you would do. And I think that this is one of the things that we now can explore with metagenomic analysis is, are there particular types of samples or particular taxonomic groups where the PCR may have been really misleading because of this variation in copy number of the ribosomal RNAs? We can now look at that with metagenomic analyses. Just like with ribosomal RNA, we've now built automated pipelines that are a hidden Markov model-based method to pull out all of the RecAs from a sample, align them to each other, trim the badly aligned regions, and build phylogenetic trees for those so that we can now do this in a high throughput manner from environmental samples. But I just think in general it's going to be nice to start to compare this across different samples for other phylogenetic markers. So the next area I want to talk about is studies. Of course, as we've heard throughout here, the next thing you want to do is look at what are they doing here. And I'm going to apologize in advance for the next slide, but people at my dinner table made me put it in. You might wonder what I was doing in this dress. My brother showed this picture at GSAC a few years ago. He didn't explain what I was doing in this dress. Ham Smith laughed until the food came out his nose, sitting next to me. I was dressed as Carrie for Halloween, which he failed to explain to people. But anyway, I'm sorry. It's after dinner. I was told I had to have some things like this. Where is Ed? Did he leave? But in terms of studying what are they doing from environmental samples, we've already seen and heard examples where phylogenetic approaches are very useful for this. So this is a figure from a paper by DeLong's group on the diversity of Proteorhodopsins that they found by PCR. After they had found the initial Proteorhodopsins, they designed PCR primers, went into environmental samples, and came up with what seemed like a large diversity of Proteorhodopsin sequences from the environmental samples. And then, of course, the Sargasso study we built phylogenetic trees. Also now, with the environmental samples, we found much greater diversity of Proteorhodopsin homologs in the Sargasso Sea data, basically because in yellow are the ones Ed had found with PCR, and the red are the ones from the Sargasso Sea. And basically, the PCR primers that he designed, even if they had been perfect, probably would not have amplified robustly across all these different groups, because again, the more degenerate the PCR is, it just doesn't work very well. So phylogenetic approaches allow you to now look at the diversity within this protein family and understand the different types that are present in that environmental sample. And with all the metagenomic surveys that people are doing, we're finding this with every gene that we look at. This is something from one of the new papers in the Global Ocean Survey, Shibo Youssef's paper on protein families. I did an analysis of some of the DNA repair genes in the Global Ocean Survey data. These in white, this is a tree of all the homologs of a gene called UVDE, UV dimer endonuclease. It's an ultraviolet light repair enzyme. This is the tree. In white are the ones known prior to the Global Ocean Survey data. These are all the new ones. And again, this is just highlighting the diversity. But to me, one of the most important things you can do with a phylogenetic approach is to start to make predictions of the functions of these genes from the phylogenetic tree. So not just saying we have a lot of diversity, but we can actually use the phylogenetic tree to make predictions of functions of genes. And this is actually where I first used the word phylogenomics, where you can take a gene family, build a tree, overlay characterized functions onto the tree, and then predict the functions of unknowns from their phylogenetic position in the tree. We never turned it, unlike with the ribosomal RNA and the protein coding stuff, we never really turned this into an automated approach. But two of the people who have are here, Kim Solander and Steve Brenner, have done this. And you can talk to them if you want to do a semi-automated approach or fully automated approach for making predictions of functions of genes from phylogenetic trees. I think that this is so much better than doing blast-based predictions of functions, that you really should look into using this for whatever genomes you're looking at. Okay, so the third topic I want to talk about is what I call the missing links, and to be different from the George Bush missing link. The missing links in metagenomic studies, which is trying to link up the who is out there and what are they doing. And basically, this has been introduced already with the idea of binning from environmental samples. Now, as an evolutionary biologist, when you talk about missing links, of course, this is what people think about. Fortunately, I have more of a metagenomics crowd here, so I don't have to really go into the explanation of why you want to bin things. And instead, I'm going to give you an example of where we've used a phylogenetic approach to do binning from environmental samples that I think shows that it's at least a useful addition to the repertoire of how you do binning. And this is a story of this wonderful insect that's called the Glassy winged sharpshooter. It's related to cicadas. So if you're from the East Coast and you're around when brood-X cicadas came out, this is like a mini version of the brood-X cicadas. The reason that it's fundable to do research on this organism is that this is the vector for Pierce's disease in grapes. Pierce's disease is caused by the bacterium Xylella. If it's found in your vineyard, the USDA will probably have you destroy the entire vineyard. And it can cause, in essence, billions of dollars of economic damage from this Pierce's disease. In fact, it's the only insect listed as a potential bioterror agent. So we didn't try to, but we probably could get biodefense money to work on this. And one of the most interesting things about this organism is how it differs from other sap-feeding insects. So people may be aware, if you do a lot of genome analyses, you've probably seen Buchnera as one of the top hits in some of your searches or in some of the analyses. Buchnera is a bacteria that's the endosymbiont of aphids. Aphids are also sap-feeding insects, and they feed on the phloem sap in a variety of plants. Phloem is the circulatory system in plants that takes liquid and nutrients from the photosynthesizing part of the plant and distributes it to the rest of the plant. It's reasonably, certainly carbohydrate-rich and energy-rich, and it has a few other things in there. The sharpshooter and its group is actually unusual in that they feed on xylem, which is much more nutrient-poor. It's what is coming from the roots, the water and the nitrogen and a few other things from the roots to distribute to the rest of the plant, but it doesn't have large amounts of other things that animals might want to feed on. And the sharpshooter is an obligate xylem feeder. It doesn't apparently eat anything else. Buchnera, the symbiont of aphids, turns out to make the amino acids that aphids need, the essential amino acids of animals that are missing from the phloem sap that the host feeds on. So this is an endosymbiont of the aphids that makes amino acids that are missing from the host diet. And my collaborator, Nancy Moran, who has worked on Buchnera for a while, was interested in whether or not there were differences in the biology of these xylem feeders. She went and did many preliminary studies before we did this work showing that there were endosymbionts in the sharpshooter group, in all of the sharpshooters that they looked at, that these endosymbionts were actually closely related to the Buchnera endosymbionts of aphids. And so we got funding and we managed to go after the genome of this, the symbionts inside this sharpshooter. So Nancy took about 200 glassy winged sharpshooters, dissected them, pulled out the tissue that she knew contained the symbionts. We made shotgun libraries from this, so it's a metagenomic study. It has host DNA, it has symbiont DNA, and it probably had, we assumed, other types of things in there that are either contaminants from the dissections, or it's not a cultured organism project. We managed a tiger to finish the genome sequence of Baumania, the symbiont that lives in this sharpshooter. And we could do all of the standard things that you could do with a genome analysis. I'm not going to talk about this. This was published in PLOS Biology a couple of months ago. You can go look at the paper if you're interested in all the evolutionary studies we were able to do now of an uncultured organism by having the genome sequence. The most interesting part was the part that I hate when I do genome analyses. I'm really bad at metabolism. And I can't get into it really well, but this really, really was interesting. So here's a prediction of the metabolic pathways for Baumania based upon the genome sequence. We think the prediction is pretty good because it's very, very closely related to E. coli, and every gene in the genome has what we think is an ortholog in the E. coli genome, so we can make good predictions of the functions of these genes. We found a huge diversity of vitamin and cofactor synthesis pathways. Those are probably missing from xylem, so the symbiont is probably making those vitamins and cofactors for the host. But no essential amino acid synthesis pathways, and xylem doesn't have any of the essential amino acids that are required by all animals to survive. So we didn't know what to do here. We figured this was definitely going to have all the amino acid synthesis pathways and maybe other stuff that was missing in xylem, and there was nothing in this genome for the amino acid synthesis pathways. Well, it turns out there were some reads left over that we could not assemble into the Baumania genome from the Metagenomic sequencing project. We didn't know what these reads came from. We tried to use methods of binning like were talked about earlier, like nucleotide compositions. We didn't do them. We didn't really know how to do them probably as well as Gluckner's group. We haven't tried his methods on this genome, but we failed to bin the sequences using measures of nucleotide composition. We couldn't get any assemblies of these sequences because all the other organisms in the sample were in a very low relative abundance compared to the Baumania genome. What we did was we built evolutionary trees of all of the predicted proteins encoded in these sequences and used those evolutionary trees to try and bin the sequence reads. And so, for example, here's one clone encoded the second peptide that we found in this one sequence read, and it groups in evolutionary trees with Drosophila melanogaster. This is probably a host sequence from some host cells that came in through our dissections. Here's another one, another clone that grouped with the Bacteroidetes group. We did this for all of the sequences in the sample, and we came up with basically 4 taxonomic groups from these phylogenetic trees. Gamma proteobacteria, these were the Baumania sequences. Arthropoda, these were, we assumed, the host sequences. This Bacteroidetes group, and then alpha proteobacteria that we think are from Wolbachia, which is a parasite of some insect species. It turns out when we looked within all these sequences for putative essential amino acid synthesis coding genes, they were all in the Bacteroidetes bin. Now this, we should have known better, I confess. Nancy had just finished an extensive ribosomal RNA survey. She did this after we had started the project, but while we were doing this binning analyses, that had shown that there was a second symbiont in all the sharpshooters in the Bacteroidetes group. It was what she thought was in a different tissue type from the one that we worked on, but it seemed to be there in all of the sharpshooters, so probably another mutualistic symbiont. We managed to get about 130 kilobases of its genome. And this, really important for our success here, was the fact that we used clone-based sequencing rather than the clone-independent methods, such as the 454 methods, because we went and took all of the clones that contained sequences that we mapped to Bacteroidetes and finished them. And that was what allowed us to get 130 kilobases of its genome from this initial binning. And we did phylogenetic trees that confirmed that it definitely looked like this was within the Bacteroidetes group. And the great thing was, you can't see this very well, that basically even in just that 130 kilobases, we found in essence all of the essential amino acid synthesis pathways. So this is a co-symbiosis. One organism makes the vitamins and cofactors for the host. The other makes the amino acids. And actually it looks like they're feeding each other some intermediates in some pathways. We never would have been able to understand this ecosystem if we had treated it all as one group of sequences rather than trying to assign them to organisms. I think it is fundamental for understanding the ecology and the biology of the system, if you can, to try and assign the reads to some organism. I'm not going to say what a species is here or what. I mean, these are just assigning them to the different cells that we had in these samples. And I think phylogenetic approaches are a useful complement to other binning methods. It turns out that if we had done more in situ hybridizations, or if Nancy had done more in situ hybridizations before doing this, we might have figured this out. This is from one of the tissue types where the symbionts are found. This is Baumania. This is this other Bacteroidetes-like organism. The cells are enormous. And in many of Nancy's studies, she had tried to filter out the eukaryotic cells to get rid of the contamination, and had gotten rid of all the sulci, including when we made our initial library. So the low relative abundance in our library was due to filtering out most of the sulci and not to its low abundance in the sample. But again, this phylogenetic sorting is really important. Now, in complex ecosystems, it might not work as well. It's going to be hard to do high throughput and accurate phylogenetic trees of every sequence from every metagenomic library that you've done. And so we've taken basically a different approach to doing this for more complex ecosystems, which is to just select, like in that Sargasso study of Phylotyping, just select a set of phylogenetic markers, build phylogenetic trees of all of those, so not every gene, but all the robust phylogenetic markers, and use those to group sequences into phylogenetic bins. And so, for example, it's in supplemental information in the Sargasso paper, so nobody's